y gracias a los que han podido venir aquí personalmente, pero estoy seguro que muchos están también siguiéndonos a distancia. Tengo el placer en el día de hoy de presentar a la doctora Jun Wen, que la conocí cuando estuve ahí trabajando junto con ella un poquito en el Smithsonian. Ella hizo su licenciatura en Central China Agricultural University en 1984 y después hizo su doctorado en el Ohio State University en 1991. Hizo 2 postdoctorados, uno en Arnold Arboretum en Harvard. Después trabajó en Colorado State University y también en el Field Museum en Chicago. Y es desde 2005 curadora del Museo de Historia Natural de Smithsonian. Ella fue también recientemente electa presidenta de la ASPT, la Asociación Internacional de Taxónomos de Plantas, y es editora en jefe del Journal of Systematics and Evolution, y fue vicepresidenta del Congreso Internacional de Botánica, que muchos de nosotros tuvimos la suerte de atender cuando estuvimos en China en 2017. Sus líneas de investigación están en la sistemática y en la biología evolutiva, y se dedica a hacer monografías, filogenética, biogeografía, etnobotánica, sobre todo la de 3 grandes familias de eudicotiledonias, que son las araliáceas, vitáceas y rosáceas. La doctora Jun Wen ha publicado más de 350 artículos indizados. Tiene un factor H en Scopus de 50, en Google Scholar de 63, con más de 14,000 citas. Uno de sus trabajos con más citas es un trabajo sobre la disyunción entre especies de América del Norte y Asia, que es que muchos aquí sabemos que hay este fenómeno muy interesante. Ella también ha colectado más de 20,000 plantas en más de 50 países, sobre todo países de Asia. Entonces tengo el gusto de invitar a la doctora Jun Wen para su plática. Thank you very much for being here.
Thank you, Marcelo. Good morning, everyone. And it's my great pleasure to come back to Mexico to visit and conduct the fieldwork. I'm very honored to give this seminar today on the collection-based integrative systematics in the new age of informatics and genomics. Can you, yeah. Oh, okay, yeah. As we know in this crowd, that systematics is the science of discovering, organizing, and interpreting biodiversity, biological diversity. And the subject has 3 sub-disciplines: taxonomy, phylogenetics, and the study of evolutionary processes. Not a, oh.
Onto the projector.
Oh, it's right here. Okay. No, I don't work it before. Okay. Well, he can advance, yeah.
Okay.
Yeah, I still needed a pointer though.
Yeah. Es que necesita el aparatito.
Oh, yeah. I mean, it worked.
Restart. Yeah.
Restart, yeah. Right. Oh, yeah. Oh, yeah.
No funcionó.
No, it works now. Okay.
Sorry.
Thank you. That's all right. Yeah. Yeah. Since the times of Linnaeus, the discovering and the describing species diversity have been an important task in our field of systematics. So in the last 15 years, if we look at the newly described species and the new combinations, by average, we have about 2,000 new species described and about 2,000 new combinations made from 2004 to 2015. This chart is an older one from the Systematics Agenda 2000, but it still makes the point right that if we compare the two columns of non-species and estimated species number, we see that systematics community has a major task to complete for the discovering of biodiversity. If you look across the major lineages of organisms, the plant kingdom actually is in much better shape. And the other organisms, especially those under the ocean, are pretty much largely undiscovered. So this really, we needed to make the public understand, because as biologists, we know, but the public generally has a misconception that the discovery phase of systematics is over. And at the dinner 2 days ago with Gerardo, we're talking about the undiscovered described species in the neotropics. My goodness, you know, the reality is here. And the herbarium at this stage house specimens which are fundamental for describing and classifying plant diversity. At the same time, in today's genomic and informatics era, the herbaria are also a very important source for genomics and for ecological analysis. So our collections really benefit not only our field of systematics, but also the entire field of biodiversity science, as well as the public. So this is one example of a old collection collected over 121 years ago, and it was described so much later by scientists. And not only major herbarium like Kew, US National Herbarium or Maxo, and small local herbarium in biodiversity hotspots, they also host very important collections for understanding biodiversity. This was a new species I discovered from a local herbarium in Papua New Guinea a few years ago, and I wasn't expecting to see new things from a small herbarium. So that this case emphasizes the importance of even smaller herbarium for our science. So in the last 2 decades, digitization has been a major effort throughout the world. And so these digitalization efforts have made vast collections and the collections data online. And this is iDigBio is one of the resources in the centered in the United States. And this, I think the data is a little outdated, but at least we have 130 million collections available via iDigBio. And the GBIF, or the Global Biodiversity Information Facility, is also another important resource for the biodiversity community. About 2 billion collections records are available through GBIF. But however, if we look at the distribution of participation of GBIF, it's very uneven. I think the GBIF data are very centered on the developed countries, and the vast collections from mostly developing countries are not available through the GBIF network, unfortunately. So GBIF really needs to encourage grassroots participation rather than charging each country with a participation fee. I think biodiversity understanding shouldn't be charged, especially those from developing biodiversity rich countries, and they are financially less advantaged. So, and right now, IAPT, or the International Association for Plant Taxonomy, has a new initiative to support small collections. And this year is actually great because IAPD is giving out another $250,000 and mostly on small grants. In the past, they provide support for each project is about $5,000. This year, it's about $10,000. So spread the word to your friends. The deadline, it's just announced. So the deadline, it's in a few weeks, yeah. And another thing is the GBIF record has progressed very steadily. So just look at the genus Vitis I work on. There are about 492,000 records available through GBIF. And even though China is not a participating country for GBIF, on the other hand, China has its own portal. That is the Chinese Virtual Herbarium portal, which is really useful. A lot of people don't know about it, but it's a very useful resource with most of the collections from Chinese herbarium available through CVH. And the CVH data not only provide the image data, you can also get data about the distribution of the specimens and also the collections, the time. Yeah, so it's quite a useful resource. And I was very delighted to learn about the Max collections available online, digitized. Thank you very much for this tremendous progress in such a biodiversity, mega diverse country. So I think this is a tremendous contribution to the biodiversity community, and I congratulate you all of making this completed this project, yeah, of over 1.3 million herbarium specimens. I think this is pretty amazing. I have been using it in the last few days, yeah. So, and yeah, to the point of digitize the collections. So this has been the utilization or applications of digitized collections have been in the infant stage. So people are pioneering the use of this so that the digitized collections have been used for not only for biodiversity research and conservation, but also for monitoring invasive species or discovering new medicine and so on. So in our lab, we actually started to use this a few years ago. So I think I'll present two of the case studies in our lab. One is to climatically characterize the Asian core Araliaceae clade. This clade is one of the largest clade in the family Araliaceae, which has about 20 genera and 900 species. That accounts for about 60% of the species diversity in the family. And this is a major radiation centered in Asia. And this clade is morphologically very diverse. So we cannot have any single morphological character to characterize this radiation as the key innovation. So we conducted a climatic principal component analysis for the family. We used the GBIF, the CVH, the data available through several different portals. So I think the digitized data are very useful for us to filtering the data noise, because at present, the data available online are pretty noisy. So as an expert, we can filter, we actually filter out about 50%, well, I guess 30% of the data are noise. So in this analysis, we utilize the 476,000 specimens. Our initial data downloaded was 683,000. And then we conducted this series of analysis. It was interesting that the most genera of Araliaceae, they occur in the climatically intermediate conditions between typically temperate and the typically tropical. So this the results. The first result would refute the well-known notion about the tropical Aradeaceae as a tropical family in the Apiaceae dichotomy of Aradeaceae and Apiaceae. So this is quite an interesting result. And secondly, the result shows this early radiation of the Asian palmetto clade may have occurred under an intermediate climatical condition compatible with the tropical mountain environment. And thirdly, the shift to the tropical conditions might have triggered the diversification, so supporting our initial hypothesis about the importance of ecological opportunities in the two rounds of major radiations in the family, in the ginseng plant family. And another case study I'm going to present is the utilization, not only the collections data for ecological kinematic analysis. And in this, we're going to use the machine learning analysis to, I think this technology has been used, has been explored to classify unknown samples into existing groups, such as this case study published in PNAS by Alex White at the Smithsonian. And also a number of labs are exploring the machine learning as a major tool to discover new morphological characters for classification. But in this case study in my lab, we would like to talk about the machine learning as a tool to study hybridization. So my postdoc, the Rich Hotel, is preparing a manuscript on this prunus diversification in Rosaceae. And so Rich filtered the herbarium specimens, and finally he utilized a little over 10,000 herbarium specimens to look into prunus diversification. And based on morphology, prunus can be classified into two major groups: the corimbose solitary inflorescence type and the tropical racemose group. And so he trained the models into these two major groups. And the idea is to test the temperate racemose group as the ancient hybrid lineage. This group, it's the hypothesis was largely based on our phylogenomic data. So we wanted to use this machine learning as a pilot study because no one has done this yet. So it's actually really interesting that the results that this was the initial basis of the phylogenetic, the nuclear and the chloroplast phylogeny showed a drastic discordance of the racemose group in the nuclear phylogeny and chloroplast phylogeny. And we also conducted a series analysis to test the hybridization scenario using the genomic data. And this group was supported to be a hybrid lineage. So then by training the data, and it is very interesting to know that the model works. The tropical racemose groups with the tropical racemose group, and the temperate corimbose and the solitary groups very well in this for these 10,200 specimens. But interesting enough, the results of the temperate racemose group goes 50% of the specimens goes with the tropical racemose, and 50% of the specimens go with the solitary and the corimbose clade. So supporting based on the morphological front using machine learning, supporting the hybridization scenario. We're pretty excited about this result. This is pretty new. He just presented that in the Barney meetings last week in Anchorage. Yeah. And so on the other hand, with these tremendous resources available online, I think it's a golden opportunity for the systematics community to develop general pipelines for taxonomic informatics. Again, this is also at the infancy, and we needed to devote a lot of informatics resource as a community through perhaps pilot efforts in major institutions such as the Smithsonian Institution. I had a very long meeting yesterday with our terrestrial biodiversity group. And one of the major focus of discussion is to look into the informatics with the digitized data we have. Our herbarium is another one which has been completed with digitization of 4 million specimens. The completion happened in May this year, so it was very recent. We're very excited about this incredible resource. And another aspect I would like to promote and emphasize is, as the taxonomic community, we should work out of the dark closet. We need to publish our biodiversity data very quickly and rapidly. And I love this, the PhytoCase model. This was actually from 2010, the first paper in PhytoCase, that they promoted this fast, linked, and open model. I think this is a great thing we should do. Many journals can follow this route. And another thing is not only promoting the international community, I think grassroots efforts can be extremely useful. I'm also a life member of this Eastern Himalayan Society of Systematics. So this EHST, this organization, published an online journal, which is very rapid. Actually, I'm writing a paper for them, which will be published in about a month. I haven't finished the paper yet. So it is a good model, and also a very important effort from the hotspots of biodiversity locally. So those people are extremely easy to work with. And I was provided with a award. So that's how I organized this talk at that time too. Yeah. So another aspect, I think the community needed to think really big. It's not like we think by herbarium. We need to think as the whole community. I have been talking about this since 2015, that our community needed a biodiversity cyberbank. So this cyberbank should not only be for data repository, we have a better, bigger task. We also need to develop tools and pipelines to handle the biodiversity data, to generate products more efficiently. And another aspect is we needed to collaborate not only among ourselves as systematists, we needed to be part of an important biodiversity effort to drive the bus, so that we should not be forgotten as the road science. I think road science has a lot of energy and life today. So, and another aspect I would like to see, this type of effort should also connect us efficiently with the public. You know, whenever we publish a new species at the Smithsonian, the public are so excited. That actually affects me as a scientist, not only discovering species, I'm also involved in a lot of projects about the evolutionary processes and the phylogenomics. I think we needed to kind of get the public engaged and support our efforts and make this biodiversity cyberbank realized as a true effort. You know, GenBank 50 years ago, they did that effort. At that time, without realizing how important it was. Biodiversity community, we got to get organized and inspire this type of global efforts. Now, I'm going to turn my attention to the second part of the talk. It's the age of genomics. You know, since the first genome published in 1995, genomics really has revolutionized all the disciplines of biology, including our field, systematics. This is the bird phylogeny published in 2014. They actually used the whole complete genome data. At the time, I was ridiculing them. I said, you know, this is too much. You don't have to kill a chicken with a butcher's knife, but nowadays, actually, the cost of getting a genome is very low. So we are actually moving into that age very quickly. So the plant community actually started with the utilization of transcriptome data. This is one of the paper published in PNAS on the phylogeny of land plants using transcriptome data. 424 genes in this study were utilized. And this was an angiosperm phylogeny, again based on transcriptome data, published in 2014. We actually, our lab started this pretty early because the Smithsonian was looking for pilot studies of using next generation sequencing tools. So I volunteered. I said, why not using transcriptomes for the grape family? So we published our phylogenomic paper actually way before the land plant and the flowering plant. We published in 2013, utilizing 417 genes to explore the deep relationships of grape family. Because we sequenced by that time 11 genes. We never really resolved the deep, it was always a calm. So this was beautiful. This was absolutely gorgeous. After we got the phylogeny, it actually makes true sense about these 5 major clades we discovered with this transcriptome data. So we carried on after the transcriptomic study with a cheaper approach, like the shotgun genome sequencing, because transcriptomes are very hard to use. You have to have fresh tissue data. At the time, we happen to have the grape, over 200 species growing in our greenhouse. So it was a really easy project. We set up the transcriptome lab and did it in a couple of weeks. It was really efficient. So, but because of a limitation of the material with transcriptomes, we really needed to use the stable DNA approach. So then we utilized following the transcriptome analysis with the shotgun genome skimming. So it's a shallow genome sequencing. We actually sequenced the whole genome, but at a relatively shallow level. But this approach is useful. We can get all the complete chloroplast genome through the genome skimming. And this was done, published in 2015. We must have done it in 2013. So the plasmon phylogeny actually supported the 5 major clades from the transcriptome analysis. And this was, we were also able to assemble 16 mitochondrial genes from the genome skimming data. And the genome skimming approach is really the power is actually underestimated. We did this pilot study in 2021. Sequencing is slightly deeper, that we can actually harvest thousands of nuclear genes from the cheap genome skimming data. I mean, each sequence is about $35 of each. So it's a very cost-effective approach to study phylogenomics and DNA barcoding and identifying species and so on. So in the last part of my talk, I would like to present a case study on integrative systematics of the grape genus Vitis using both informatic and the genomic tools. So the genus Vitis, it has about 75 species with 2 major centers of distribution, about 35 species in the New World, and about 40 species in Eastern Asia. One species, very important one, the wine grape, is from Europe and West Asia. So actually in this first study of Vitis, we used genome resequencing. This was because we had a Vitis genome, and the resequencing would be really easy. The data analysis part would be pretty cool, and we were able to utilize over 2,000 single copy genes by comparing 5 genomes already available in gene bank. So this was a pilot study we thought would be nice and cool and easy to pursue. And so we did it. And the result was pretty interesting that the vitis, the genus was supported to be with 4 major clades. And so the Muscatinia clade, it has only 2 species, one in North America, one in Mexico. And then another North America clade of Vitis subgenus Vitis, and then a European Vitis vinifera, and then the Eastern Asian clade, a large Eastern Asian clade. So this was very congruent with actually geography, the phylogeny. So based on our biogeographic analysis based on this genome resequencing phylogeny, the first one ever produced for Vitis, supported that the genus, the grape genus, originated in the New World and then migrated through Europe into Eastern Asia. And then within Eastern Asia, it actually had a north to south, within Eastern Asia, a north to south pattern of migration. So that was kind of very interesting for analyzing the Vitis genus at the generic level. So because at the time when we pursued the analysis, genome resequencing was still relatively expensive. So then we switched in the last few years with the target enrichment or the HiBSeq approach. And this approach does target certain genes we know. So in this case, we had 229 genes from our transcriptome data. So we targeted those and designed the probes using the genic data as well as the Vitis genome, designed a set of probes to analyze the Vitis evolutionary history. And so at the first, we look at the Vitis, the Asian Vitis, because that's a monophyletic clade. So we look at this, and for the first time, we recognized 7 major clades within Eastern Asia. And also we analyzed the phylogenetic diversity, and within Eastern Asia, the phylogenetic diversity in the southern part of Eastern Asia is much higher than in the northern part of Eastern Asia. And we also utilized the vast digitized collections from GBIF and from CVH to analyze, for a niche modeling analysis, to study the spatial diversification of the Vitis genus. Very interesting, you know, the different major clades we recognized, I mean, they show very similar spatial diversification patterns. So this was unexpected but interesting results for us. Is this parallel geographic diversification of different clades? They mirror each other in the diversification process of the genus Vitis. So, and then we analyzed each of the 7 major clades in terms of the drivers of diversification, and the detailed analysis of these 7 clades support ecological and geographic isolation as the major drivers for diversification for Eastern Asian Vitis. And also there are some hybridization events, especially in northern and central China. So that's another analysis. We used the network analysis to look at not only the tree of life, but also the network within Vitis. This is the network within Vitis, the Eastern Asian clade of Vitis. Again, there are several hybridization events in this clade. Yeah. And so to summarize this small portion of the Eastern Asian grape studies, so the comparative analysis integrating the phylogeny, ecology, geography supported that the diversification of major clades, they utilized the different ecological and geographic space parallelly, so which is a very interesting model for understanding the super rich diversity of Eastern Asian Vitis. And now I'm going to the last few minutes, I'm going to talk about some new results of the evolution of VTs in the New World. And so this again, we used this HyperSeq targeted enrichment approach. We targeted 318 single copy exon regions. And so we sampled the genus broadly, but we emphasized the New World. But because of Eastern Asian grapes, it's a monophyletic clade. So we selected a few in this analysis. So this nuclear phylogeny based on the 318 exon regions support 4 major clades as well. And the first clade, it's the Vitis, it supports the subgenus Vitis and the subgenus Muscardina, each as a monophyletic group. And then within subgenus Vitis, there are 3 major clades. So we have the Eastern Asian grape, and then we have 2 major clades in the New World, New World 1 and the New World 2. New World 1 is largely distributed in the western part of North America, and the New World 2 is largely in the eastern part of North America. And note that the closest relative of Vitis is the New World Amplocetus centered in Mexico. So this group of New World Amplocetus, it's the closest relative of the economically important Vitis, but it didn't form a clade. It formed a paraphyletic grade in this case. So this was very puzzling. I think it's another reason I'm here too. Yeah. So the New World Amplocetus consists of about 5 or 6 species, and they are morphologically also very similar to Vitis, but it differs from Vitis by the character that the ventral infolds, the holes in the seeds, are much broader. It's a cup-like. And in Vitis, normally it's linear, narrower. But if you look across these 4 species examined by Steve Manchester's lab by one of his graduate students, you see this vast integration from very cup-like to much narrower cups. So that integrates into more like Vitis. So in this case, I think in 2018, actually I proposed the Wen et al. We published a paper on the Vitaceae classification. We proposed the merge of the New World Amplocetus with Vitis. With this merge of this grade into Vitis, Vitis sensulato and Amplocetus sensu stricto, they are each monophyletic, so which seems to be great. And certainly the phylogenomic results also support such, both nuclear and the plastome support the merge. And interestingly, in the plastome phylogeny, there is a little incongruence that one of the species, Amplocetus from Mexico, is embedded within the genus Vitis. So once again, both nuclear and the plastome data suggest the merge of Amplocetus from New World with Vitis. But on the other hand, we needed to really figure out what have happened of this deep incongruence between nuclear and plastome data. So that was a little too fast. So we analyzed the nuclear data with different analytical strategies using concatenation as well as coalescent methods. As you can see that the nuclear data with different analytical strategies, they showed deep level congruence, but at the tip level, they showed incongruence. So that perhaps indicative of hybridization at the younger level. But on the other hand, if we look at the nuclear and the plastome phylogeny, it's unbelievably discordant. that so strong cytonuclear discordance has been detected at both deep and the shallow levels. So we conducted, I mean, this was very interesting. So we conducted a series of analyses to analyze this, to conduct the hybridization analysis within New World vitis. One of the deep incongruence in particular concerns the three major clades within vitis subgenus vitis. In the nuclear phylogeny, the Eurasian clade is nested inside a nuclear grade. But in the chloroplast topology, you see that the Eurasian clade is sister to the New World clade. So this indicated deep hybridization. So we conducted many analyses. One of them was the FICO analysis to check on the hybridization scenario. But interestingly, the FICO analysis indeed support this hypothesis. So the FICO analysis actually suggests more nuclear genes supporting the plastome topology. However, the difference of the log-negative scores of the genes favor the nuclear topology. So these results clearly support this deep hybridization event. So we propose that the New World 2 from largely in Eastern North America as a likely hybrid, Asian hybrid of vitis. Yeah. And we also conducted a series of network analysis using the strategy SNAC. Because SNAC is at present is still computationally very intensive. So we cannot really efficiently analyze more than 30 accessions. So in this case, we designed 6 major sampling strategies to test extensively of this hybridization scenario within vitis, emphasizing the New World. Again, lots of hybridization went on. Two of them was completely out of my expectation, which actually makes sense now. One was the deep hybridization of vitis subgenus muscadini. That's the one with only 2 species, one in North America, one in Mexico. So this was suggested to be a Asian hybrid at the deep level. So that explains the jumping of one of the Amplocissus species into vitis in the chloroplast phylogeny. And another one unexpected was there was an endemic species from the peninsula Florida called vitis shuttleworthii. I mean, the species was, I always thought it's very close to one of the species in Southwestern North America. Never doubted about it. But the analysis suggested that it is not only close to that species, it is also showed a hybrid edge with a neotropical clade from Mexico to Costa Rica. I mean, the tropical environment in Florida is agreeable, consistent to this unexpected hypothesis. Now, when I look at the morphology makes sense of this hybridization scenario. We just never thought that way. So the analysis really gives us some new insights into the network. It's not a tree of life, it's really the network of life, especially in this complicated genus vitis. So we further pursued biogeographic analysis of the genus vitis and also the dating analysis too. Our analysis support a very deep origin of the genus vitis into the early Pliocene. And so our analysis supported the Pliocene origin of vitis in North America. At that time, North America was covered by the thermophilic boreotropical flora. And by Oligocene cooling, vitis retreated southward into Central America refuge. That's why we see these very unique lineages in Mexico, Costa Rica, and so on. Yeah. And early to mid Miocene, VTs moved northward and radiated across North America. And our analysis also suggests vast reticular evolution or hybridization in this process. And during the late Miocene and Quaternary cooling, VTs migrated out of major refuge and diversified across North America. And so the boreal tropical refuge and these extensive interactions are supported as the mechanism to account for the diversity patterns of North American VTs in three major areas: Central America, Western North America, and Eastern North America. And also, I'm not presenting to you, but I want to summarize a little bit about our last decade of work in systematics of Vitaceae. And we proposed we have now classified the 950 species into 5 tribes and 16 genera. And I want to point out one interesting fact that all those genera, even some of them were in Vitaceae before, but the generic concept is very different now. So we have 6 genera. Out of the 16 genera, 6 of them have been redefined, and 1 genus resurrected, and then 2, at least 2 new, I think I would say 3 now, okay, genera are new to science. So really vastly advanced in the last 15 years on systematics, and Vitaceae is only one of the examples. One of the advances we talk about because of, I'm the Vitaceae, I'm interested in Vitaceae, but many other examples exist. So that it's really a pretty exciting time for systematics at the age of genomics and informatics. We can really analyze lots of data from the biodiversity perspective and the tree of life, or really network of life perspective. Yeah. So another thing I want to say that it's our field, it's really in a new age of modern synthesis with this vast data and a new age of discovery, because we really discovered a lot of genera and species through this process. And even though our systematics with 3 sub-disciplines, but these 3 sub-disciplines are so much better integrated via informatics. So our field is highly integrative today. So I'm really excited to think about this now, the age of a new modern synthesis for systematics. And so we are in a new age of discovery and synthesis in systematics. And I want to last summarize that herbaria and museums are treasure troves for undiscovered new species, and also are such important resource for genomics and ecological data. And second, there will be major realignments of genera and infrageneric taxa, as well as higher taxa based on phylogenomic evidence. And it's a golden time for collaboration in the systematics community. Finally, it will be also an exciting time for species-level systematics in the next decade, using integrative systematic approaches, integrating morphology, phylogenomics, ecology, and geography. And the last, well, not really second to the last, I still want to emphasize that our age of discovery is not over. We really need to train our next generation to explore the vast unexplored regions. So this cannot be the importance cannot be underestimated, because I'm so afraid our new generation can sample quickly and get the results and publish. Go and explore. The field is the vast laboratory. Each time when I go to the field, I really found tremendous questions, and I get excitement and found new questions for exploration. Yeah. So this is the last slide, is that As a systematist, we really need to develop systematics broadly in the biodiversity science. It's the time we needed to argue our importance and our contributions to the community. I mean, our vast natural history collections would always serve as the foundation of biodiversity science. And also one of the things we need to emphasize, and we don't do very well, is to actually promote our science in the public. So then our science will be better supported. And I was so excited to hear about this new museum in Vietnam that, you know, this guy, Mr. Carlos Slim, visited the Smithsonian, and he was inspired by our museum, and he came back with this tremendous idea of establishing this new science museum in Vietnam. I think this is inspiring. This is really the outcome of bridging our science with the public. And we can not only do that by reaching out to them, we can actually make our science somewhat more practical, such as, you know, we can study a lot of, I'm also having a strong interest in Isotopy. I have a long-term collaboration with the Food and Drug Administration in the United States, so that we have been collaborating with them for close to a decade. So I think I want to emphasize that we are not only systematists, we are biodiversity science. We can help the human society. We can link it with them. We can excite them with the rich diversity and help the children to move away from the computers and from these little apps. And they need to go look around of the green nature. I think it's very inspiring. Thank you very much. Yeah. Yeah, any questions in Spanish is okay. Marcelo can translate. Yeah.
Sí. Yvonne. Ah, sí, perfecto.
Hello, thank you for your talk. My question is regarding your case study with Prunus and the machine learning study. In that one, you first identify a few clades, and then at the end, I think your inference was that the temperate clade, I think it was called, I don't remember.
Yes.
You said because some of the specimens in the temperate clay went up and some of the others went to the other clay, right?
Right.
Some of the members.
Yeah.
And I think your inference there was hybridization. You said that that was clear evidence of hybridization. But why would it be hybridization and not something like just a mischaracterization of that clade? Maybe there are other entities within the clade. Maybe there are two clades inside that clade.
Yeah, it's thank you for the question. And I think, yeah, the hybridization hypothesis was also, it's a more integrative analysis because we analyzed the entire genus Prunus also with the final genomic data. And the final genomic data had showed a very strong signals of hybridization of the temperate rhizomous clade as an Asian hybrid between the tropical rhizomous clade and the temperate single flower and ambulate flower group. So that's the initial hypothesis was proposed based on the results. And then at the time, because machine learning is such a new effort, so we thought this will be an interesting case to test how the specimen, the morphology data would go. So then we trained the model with 10,200 specimens, and that did not include the 1,200 specimens of the temperate rhizomous because that's the target. Mm-hmm. We trained the model with the two major groups. And then we analyzed the 1,200 specimens with in the broader scheme of the two major groups. So the machine learning data support the hybridization. Yeah, that's because of the integration of the data. Without that, it could have like a deep, maybe there may be other mechanisms, but the best mechanisms we can arrive at based on both machine learning and the genomic data would support the hybridization scenario.
I see. Okay.
Very good question.
Thank you.
Excellent
I have another
question.
one.
Okay.
When you compare, it was, I believe, chloroplast phylogeny.
Yes.
It was a chloroplast phylogeny here, and I believe it was a mitochondrial phylogeny here.
Okay.
I don't remember. I might be wrong, but it was a chloroplast phylogeny here. And you also mentioned that, well, you said that they were very, very incongruent.
Oh, that's
right?
the nuclear. It's
a
the
nuclear, right?
The,
That's right.
Yeah,
and then the chloroplast.
On the right.
Right.
Yes.
Well, what I noticed in the chloroplast phylogeny here on the right is that it had very little phylogenetic signal. The internodals were very, very short, and there was no much resolution. So wouldn't be maybe more appropriate might be the word, or maybe be more cautious about the signal that these kind of genes are providing, and maybe they are not as reliable to phylogenetic inference, not necessarily pointed to, well, there is incongruence there, but it's not real incongruence in the sense that two processes are happening. Maybe it's that one gene is just not very good. Just like you pick a morphological character and it's just full of homoplasy, and then you begin making stories out of a character that is just not very good.
Yeah.
You know
well,
what I mean?
if you look at the phylogeny of the chloroplast, in particular at the deep level, we look at the 4 major clades, they're all 100% supported. Very strong support. And also the FICO analysis we conducted support this hybridization scenario as well. Some of the tip loads, I think, could be linear sorting and others, but there are major hybridization events supported by additional analysis. Not only just chloroplast incongruence, but it's utilizing the analysis of nuclear data alone, because we are looking at 318 nuclear genes. So not every species trait of that telling us the same story. So we analyzed each of the 318 genes very carefully. So hybridization is not based on the incongruence. Hybridization is based on the vast analysis of the nuclear gene data as well. FICO analysis is one of them. Yeah.
Okay. Thank you.
I have a question here.
Just comment that thank you very much. This brings a greater responsibility of taxonomists, because there are so many studies now that really overestimates the results, because they just checking platforms, databases, but they don't come back and check with the taxonomists.
Yeah.
It's because there are a lot of mistakes in herbaria.
Yes.
And that brings you to overestimation of those results at the end if you are not going back and check with taxonomists.
Yes, I think it's very important for taxonomists working with, it emphasizes the importance of collaboration. For our RLAC analysis, we found that 30% of the data are noisy. We started with 680-some thousand records, and because of the data are digitized. So that's why I wanted to see how important this is. So me and also my students as an expert can really verify the identity of those. And then we used the 60% of the data after the filtering of the noise to come up with the evolutionary analysis. I think this is very, very important. You made a great point about,
because this is a major risk.
Yes.
If you don't go back.
Yes, I think this is to truly, advocate the value of systematics. We shouldn't be forgotten. We are extremely relevant.
That's why the last of your slides shows the human division or human attention, shall we say that? And this brings the taxonomist. But throughout your talk, I can imagine what Yvonne saw about chloroplast and nuclear. They are inherited differently in plants. But when you are working with prunus and vitis, you're working with plants that human beings have been dealing for thousands of years. And could you feel that some of those hybrids are really made by this moving plants to one side and the other? And if this is so, then some of those hybrids are new, well, recent, but also it brings the importance of pollination studies.
Yes, that's a great point. Yeah, I think our study, we indeed encountered the hybridization of vitis in the California, the Central Valley, in the Napa Valley, so that we have the prunus vitis californica. I think the most of the vitis californica in the Napa Valley, they are not pure. And only the northern, but the good thing is now it's really, if we look at this problem 10 years ago, impossible, big headache. But today we actually can piece apart how the hybridization happened. We can clearly see the pollen came from the wine grape. So that, I mean, it happens, but it actually didn't happen as extensively as we would envision, because of the processes with these genomic tools, it is actually pretty powerful of teasing about reticulation events.
Yeah,
yeah.
I'm trying to remember, because vitis, the European one, the cultivar.
Yes.
In fact, it uses roots from the New World vitis because they are more resistant to disease.
Yeah, that's another interesting area to study. One of my former students, Alice Miller at St. Louis University, she is looking at a lot of genomic exchange between the scion, at that, the grafting point of the stalk and the wine grape. Yeah, so that's virtually the frontier of that type of analysis. Yeah, so it is a very interesting point that how at the genomic level, the gene exchange of those things. We're going to have a major workshop in November in the Oak Spring in Virginia on a grape. It's a comprehensive discussion of scientists like myself and also a lot of folks in the industry. So to talk about some of these, the frontiers, the challenges, and the future directions of these interesting problems you just mentioned, at least 2 of them. Yeah. So,
okay, thank you very much.
Yeah, great questions. Yeah.
Thank you. I enjoyed your talk very much. It was very inspiring. I have a very precise question. You didn't tell us much about the Asian diversification, but you presented a slide in which there were 2 phylogenies, one from a concatenated database and the other one using coalescent approach. And it was interesting to me that in other instances, there are like overall congruent patterns and the tips are mixing up. But in that particular study, there was a whole yellow clade that with the concatenated approach was near the base of the phylogeny, but with the coalescent approach, it was very derived.
Yeah, I think it's the same tree.
Something?
It's the same tree. We switched.
Okay,
because,
it is switched.
Yeah,
okay.
It's the same topology, actually. It's
So,
when we generated a tree, I think it was from a different from a different paper. Yeah.
All
So
right.
I think, yeah, we didn't. So, but the topology, the place of Asian clade,
All
it's
right.
congruent. Yeah.
Perfect. So
Yeah.
it wasn't strong incongruence among methods of analysis.
Not
It was
for
only
the
how you
nuclear,
painted the
not
tree.
for the nuclear data, but
Okay.
there is a strong incongruence between nuclear and chloroplast. Yes.
Thank you.
I have a question here in the website. So Fernando Cervantes asks you if hybridization is a major issue in your interpretation of speciation in vitis. Did you include research data of vitis, or species of vitis on hybridization from cytogenetics and karyotyping?
We did not in our efforts. The chromosomal level in terms of chromosome number, vitis, subgenus vitis, we're not experiencing any polyploidy or anything. Yeah. So that I think, but at the deep level, I think it's still worth to look at the cytogenetics of the, because we did detect the very deep events, at least the two of them. So that it's yet to be studied. Yeah. We have not done that yet. Yeah.
Perfect. Ok. ¿Alguna cuestión más? ¿Alguna pregunta más?
Oh, thank you very much for your wonderful talk. I'm very happy to be here and listening to you. It's so inspiring. I would like to ask you, well, I think that usually people ask about well, you already talk about this and the need for collaboration with people that knows about groups, taxonomic groups, and the need to be cautious when we interpret our results and all this. But I would like to ask you about the future of the field. I really would like to ask you about something more crazy, for example, the future of machine learning. I really would like to know, what do you think, what will be possible in the future for this technology, and the, yeah, what machine learning can do for taxonomy and systematics. And I would like to ask also about, I don't know, what do you think is necessary still to do in phylogenomics, for example, with dealing with heterozygosity, polyploidy, and all this. Thank you very much.
Yeah, that's yeah, both areas of the questions you ask, they are advancing tremendously. Yeah, I think the machine learning front, we're still at the very early stages, so that we need the major institutions like UNAM, I think would be great if people can really get organized and support not only getting the data available, but also make the data meaningful. So this type of the pilot studies, I suggested there are very few of them available because it's a brand new field advancing gradually. And at the Smithsonian, we realized the urgency and the need. So we are actually creating a informatics, what did we say? It's not a group, think tank, yes. So we're creating such a think tank to explore, to help drive ideas of how we should move forward. So I don't have the exact answer for that. I think it's a great thing. You know, if we knew what answer, our discipline is boring. You know, I think it's great. We don't know the answer. And secondly, in terms of the phylogenetic, the future of phylogenomics in dissecting polyploidy and hybridization, I think that future is very bright. I think at present, I think the limitation is the computational power of programs are still very limited. So like, for example, for the VT study, we really had to design 6 different data sets to analyze these events. So we really need to advance the informatics. And another thing is, I do think the field work and the detailed understanding of the morphology is very important. I think the advantage of VTs or prunus, I'm really, as the PI in my lab, I'm extremely familiar with the variation. I take students out every year to the field for weeks of field work to look at these things. I even took undergrad interns with me to the field. So I think it's kind of nice that some of the questions are coming from the morphological variation. Sometimes with this odd thing, I said, oh my God, this looks, and then we come using the tools we have in the laboratory. So systematics is truly integrative. The future is going to be very integrative. If we only use the results for machine learning, we may be misguided. I think we have to really look at the morphology, the genomics, the phylogenomics, the ecology, and and putting the scenario together. One of the slide, one of the story may not necessarily to be correct. Yeah. I hope I answered your question. Yeah.
Ceci, ¿hay algo más? ¿Alguna pregunta más? Then thank you very much, June. We have cookies and coffee here outside.
Thanks for coming. Yeah.